The real challenge of big data ultimately comes down to the big. And for any individual cancer patient, we might look at 3 billion different characteristics.
It's difficult to get sufficient patient samples in order to prove a particular point.
Lack of patients on clinical trials certainly limits the number of samples that we can work with, but it also is optimizing the use of any single sample that we get from a patient.
Okay, so these are some of the screen cell filters that I isolated CTCs from yesterday from the expect patients. When somebody agrees to take part in a trial, you want to respect their samples. We were running their serum, their plasma, and their whole blood samples. So we want to get a huge amount of information back.
When patients are generous enough to donate samples, we need to manually cut sections. We use our laser microdissection and harvest the tumour cells that we're interested in. In parallel, we're interested in metastasis. We collect liquid biopsies. With next-generation sequencing, we've now got to the point where it is really, really sensitive. It's a form of confirmation for us at this point so that they can be successfully implemented in the clinic down the line.
The technologies are evolving incredibly quickly. We used to do sequencing just 5 years ago that might have 30% error. Today it has 2%. That's an immense change.
One of the major challenges that we face now is dealing with data. We need people who are sufficiently skilled with computer programming and bioinformatics to be able to do that for us and help us.
It's a lot of data about each individual, but we need to collect it on thousands or tens of thousands of patients to be able to have the statistical power to make predictions and to really be able to accurately refine care pathways.
The other difficulty with data is it's limiting if we're trying to accumulate a large number of samples. It requires us to biobank, but there's no funding typically to sustain that bank. And so the next researcher who comes along, either to ask a follow-on question or a similar question, they will get their funding and then they start from scratch.
So there seems to be something here, but if you zoom in...
The biggest problem with big data and international collaborations is sharing the data.
In terms of solutions, cloud computing is a big one. There has been work done to build containers of pipelines and put them up on these cloud servers such that groups can upload their data and then they can pull their data back down.
The field of cancer big data is really evolving to be more open. We don't keep things privately, whereas a decade or two ago, that was everything.
What we're trying to do is get rid of some redundancy. You don't want someone sequencing 100 genomes in China if you're going to sequence the exact same 100 in Canada.
International partners is so critical, and one barrier that needs to be broken down is that we need to embed research in the clinical community, and I've seen that in the best centres that I've been involved in.
We need to move away from the idea in some cancers that we're just going to cure it. And we need to accept people can live well, notwithstanding they have a chronic disease. And that chronic disease, much like diabetes or asthma, happens to be cancer, but we're dealing with it.